And what I'd like to do is I'd like to introduce Jiri Aubrecht. He's one of the co-directors for the Predictive Safety Testing Consortia. He's also an executive director at Sarepta Therapeutics. I guess that's your day job, Jiri, when we're not driving you in PSTC. So I'll turn the floor over to you, Jiri. Thank you.
Thank you, John Michael, for the nice introduction. Let me start my talk. Hopefully I'll get these moving. Okay, so I was asked to provide my perspective on importance of the PSTC to the industry. And let me just start with saying that we all strive to really revolutionize the drug discovery and drug development by applying a patient-centric approach to drug development. It's all start with the patient, and from the patients and understanding the patients, we try to identify the targets and develop tools and therapies and close that circle of the forward and reverse translation. As opposed to what we've been doing for decades is for drug development, it was called from bench to bedside, starting with the target and then developing, going through series of studies from non-clinical, in vitro, non-clinical, and at the end up in clinic. So if we want to really develop drugs using this patient-centric approach, we need biomarkers. The biomarkers are written all over it. So the biomarkers promise us really to better understand the patient and the disease, and make the drug development really efficient. And what we also not really talk that much, but I think it's really important piece is to improve access, using biomarkers to improve access of patients to the therapies, and really develop the personalized type of medicine which we all strive for patients. So we accept that this is the way for drug development and to improve care for patients. So and the biomarkers are really central to it. There's challenges to it, and they were recognized by the Critical Path Initiative, which started in 2004, as you heard with that meeting in DC, and well understood that we need drug development tools, we need biomarkers, we need a biomarker qualification, we need qualified biomarkers, we know how to use them. And for me, the biomarker qualification, it's really building scientific consensus among all stakeholders how the biomarker is going to be used. Okay, understanding its performance and understanding the caveats, and get the consensus around it. That has been done empirically without a biomarker qualification process over years, and it take decades to get to a point of getting a biomarker which is universally used to a context of use. And it's built by reaching a consensus by looking at variety, by people publishing papers, researching, and then all comes together empirically together to which codify the use of the biomarkers, and it's used by the scientific community. So the biomarker qualification is basically a process which allows us to reach that consensus faster and give it a form. And the other challenge is the resource to develop the biomarker data sets. And you heard it from the speakers, from Amita, from Nick, and everybody, and John Michael, is there's no single entity which can develop a biomarker and bring it to the use. So it really needs that collaborative approach. And of course, in the beginning, there was lack of coordination of that, and who is going to do it. And there where the Critical Path Institute and the PSTC play a crucial role. Okay, so now let me just kind of give you on the next slide, kind of when I'm talking about the biomarker qualification and the process and how that is done. I'll point you to the paper which John Michael mentioned in Science Translational Medicine, where we work together with all stakeholders, companies, regulators, the FDA folks, CROs, academics, to develop a evidentiary standards or develop a process at which we will be assessing these biomarkers. Okay, so and kind of a roadmap for building the scientific consensus on the use. So it all starts with the defining the need. Of course, we're not going to develop biomarkers there is not a need, and which follows with assessing a risk and benefit of that biomarkers, and how if they are a risk, how we actually mitigate them. And that comes to a point of really understanding what evidence do we know to show what that context of use, or I mean the use of that biomarker, it's what do we need to satisfy or to get the answers on that benefit and risk ratio, how we assess it. And then comes the process at which the biomarker developers are working toward getting the data to satisfy or to answer the questions on the biomarker performance. And it's a constant discussion with the with all stakeholders, including regulators. And once we accumulate enough data, that risk-benefit ratio can be answered for a population of patients in that particular context of use, the regulators then reach the decision to qualify that biomarker and they qualify that context of use at which the biomarker is then used across programs. So if we think about it, you know, the understanding of that risk-benefit ratio changes with data, changes with what population of patients it's going to be used. So, of course, to get the qualified context of use, one require a lot of data for the scientific community, not only for the regulator, but for the drug developers the same, and the academics to really be comfortable in how the biomarker is going to be used. Okay, so but that doesn't prevent the sponsor, the pharmaceutical company, the biotech company, to go to use the biomarker under their specific INDs, because then you can think about it that use is much more narrow in a very specified patient populations. So one can discuss that with the agency and get that biomarker accepted under the IND and use it in that specific program, and that's a huge value. And going through the process of the biomarker evaluation or validation or qualification with the PSTC, with the consortium, with the collaborators, allow to accumulate data that one can then use to support the accepted use for individual program. And I think this is a very underappreciated value or impact what PSTC has on industry. And on the next slide, I'll give you an example. And we've been talking about that example. Let me just look at this here. We've been talking about this example quite a bit, and the case study of the glutamate dehydrogenase is my favorite, and how that actually pan out if you look on the left side with the process. So the unmet medical need in this particular case is a detection of onset of liver injury in subject with underlying muscle impairments. Can be any of these dystrophic diseases or anything like that. And then our goal was to develop a biomarker which would address the lack of specificity of ALT, and we will do it with GLDH. And as you know, most of you know that the alanine aminotransferase, it's used commonly for detection of liver injury, but because it leaks from the hepatocytes. but actually alanine aminotransferase was first discovered as a biomarker of myocardial infarction, because a significant amount of ALT is actually in skeletal muscle or in heart. So, so once you have a underlying muscle injury, the fluctuation of the ALT data levels does not allow you to detect the onset of liver injury. How did we go about it? In terms of the GLDH, and John Michael talked about it, we basically pioneered an approach where we use a disease to approximate the drug or organ injury. And we also use a acetaminophen overdose patients, and assembled a large data sets at which we were able to establish the performance of the GLDH. And those were published, the first series of a large data set, like the first one at Tucson, included over 700 subjects. With plus one, we've got 400, almost more than 400 subjects in all those studies, where we show the performance of the biomarker. So, and as we were doing this and working with the FDA on what that evidentiary standard, how we satisfy the evidentiary standard, which are described in that paper in translational medicine, Science Translational Medicine, we went to the FDA and proposed that GLDH to monitor for liver injury in drug development under IND. And which was accepted, and based on what we know from other members of consortia, that it's been required or been accepted not only for one company, but for other companies too. So that's the value at which you can actually pursue or put a biomarker for decision making, and it's a direct impact of what has been done with the consortium. All right, so and on the next slide, just want to tell you or point out that the drug development is it's not only the destination for the biomarker. And as we know, the biomarkers for drug safety were basically developed from biomarkers which were developed for diseases. So now the circle kind of closes up by bringing these biomarkers into a disease area, because as you can see on those 4 panels, yes, when we characterize the biomarker, the GLDH, and if we define the cutoff and the clinically important levels at which the physicians or clinicians can use them for their decision making. Now you can use them for drug development, and that's on the panel number 2, where this is an old slide and our first patients with Duchenne dystrophy and the ALT, the red line fluctuates. You are not able to see any, if there would be an onset of liver injury, you're not able to see it. And the blue line is the GLDH, which is completely unchanged in those patients, which then provide the evidence of lack of liver injury in these patients, or evidence for safety, liver safety for that particular medication. But on the slide, on the panel number 4, you can see, and that was published in that PLOS paper, where we look at patients with rhabdomyolysis and time courses, and actually we're able to diagnose liver injury in those, onset of liver injury in those patients, which currently no other method can do that. Okay, so that's kind of a it's has a potential to help a general practice and patients with a variety of conditions. And as the last one, we embark on studying about 4,000 subjects in large lifestyle trial, and look at a correlation of GLDH and ALT with diabetes, body mass index, genders, and so on, other factors. As you can see, there are p-values on that. So as what we found, what it's commonly known that diabetic patients have higher values of ALT. Usually, people with a large body mass index have larger levels of ALT than people with lower body mass index, and so on. And the GLDH is largely unchanged. So, which then leads to kind of hypothesizing that we might have GLDH might be even a better biomarker of liver health than ALT. And there is a substrate for many studies of others and clinicians to understand, how GLDH can be used in chronic liver disease, NAFL, NASH, and so on. So this is kind of to demonstrate that the work of the PSTC and working in the space of drug-induced or chemical-induced injuries provides a benefit to clinical practice. So my last slide, I would just kind of summarize what the importance, what I see the importance of the PSTC to the industry. And to me, what jumps first and the most important, impactful piece, which we have done, and I feel privileged and proud to be part of, is the development of evidentiary standard for biomarker qualification, and which led then to the guideline, and not only for the clinical, but also for the bioanalytical requirements. That's another piece. So those are important roadmaps of how to actually do a biomarker research, and it goes well beyond of qualification. It's really a roadmap of if you do any biomarker evaluation or biomarker research. So how you approach that. So the impact I talked about and I point out, it's about the regulatory acceptance. It's about learning from the regulatory folks at the agencies, from the academics, and working together to get the data which supports the acceptance and building that scientific consensus, which comes to the qualified uses after the qualification, which an example could be the acute kidney injury markers, or as an example I gave you on the accepted use for the GLDH in clinical trials under INDs. And that's a huge impact. So again, you heard from everybody and all those speakers that providing environment for collaboration is really important among all stakeholders. The way that we all learn from each other. My experience with interaction with everybody is absolutely positive, and I think nothing would be possible. We would not be able to have a GLDH, AKI biomarkers, anything without that. Another important piece is really resource and risk sharing, and that was touched on many times. In this pre-competitive space, sharing samples, sharing ideas, sharing assays, sharing knowledge and know-how for calibrators, for assay performance that we have harmonized across companies. Across the number of laboratories, it's absolutely important. John Michael talked about the database. That database, I think it's going to be hugely important once we start, getting a critical mass of data in it. And I can see that this to be a game changer in application of biomarkers in the industry, because it will be accessible to everybody to evaluate the performance of markers. The other thing I believe it's important on a personal level, the career development of members, because that gives you and all the researchers across companies working with others, publishing papers, and really developing their scientific skills. And I think that's essential, and it's important for the management of those companies to recognize it. as a important tool. It should be an important tool in career development of participants. And then the last part, I want to kind of say tell you my thoughts on what whether it benefits the large companies or small or so. As you can see, the membership of those companies, they are largely large companies. And I had the opportunity to work for large companies and small companies. And I see the benefit actually for all of them, on all sizes. And I would even argue that the smaller companies would benefit a lot more from PSTC than the large one, because obviously there is a lot less manpower, and they might work more in isolation, and bringing them together and working under the umbrella of the consortium helps tremendously in terms of understanding biomarkers and how they can apply them to their particular programs. That they learn the science through the consortium, but then how they apply it to support their INDs, support their clinical programs, that's all up to them. And I think it's enormous benefit. So with that, I'd like to conclude my talk, and thank you very much for listening.